it's a pleasure to be here in the Natural History Museum, which is, of course, a treasure for the University of Iowa. And thanks to everybody who came, friends, family, students, et cetera. And so I'm going to tell you about the ongoing work in my lab, which is about sex, of course. And so I changed my title. So the title that I had on the flyer was It's Sometimes Strange Evolutionary History, but of course it seemed, in retrospect, a little bit more appropriate to call it natural history, which are basically the same thing. Natural history is indeed evolutionary history. And as an evolutionary biologist, I thought I would point that out. So this is a snapshot of the people in my lab and a guest. And you can go take your picture with our guest. This is about a year ago, so it doesn't need a lot of updating. There's a couple of new people, and there's some people that have departed. But for the most part, this is the assembled cast of players that have contributed to the work. Including some people in the audience, and the person who ran my lab while I was on sabbatical, Cindy Brochu, who's in the audience. So she deserves a lot of credit, and lots of other people in the lab have contributed to the work that I'll tell you about. And this lists all those people, including people that are current, and a lot of the work that I'll present came from a former graduate student in my lab, Banu Malik, who's gone on to do postdoctoral work in a couple of different places, and she's doing very well. But the work I'll present is very much a group effort, and a lot of people have contributed, from postdocs, graduate students, lab technicians, undergraduates, even some high school students have contributed to some of the work that I'll tell you about. The money is important because the work that we do isn't cheap in some ways, and we get support from the National Science Foundation, the National Institutes of Health, and our local agency, the Carver Trust. And of course, you all have contributed to the NIH and NSF funds indirectly in terms of your tax dollars, so I appreciate that. So I have some great colleagues who have collaborated with, including Bob Malone at the University of Iowa and Maureen Neiman, who's in the audience. I won't talk about our work with Maureen. And a lot of the work that we do is based on work that's existing out in the world, which are genome sequences. So in the last 10 years or so, there's been a lot of effort put forth in sequencing genomes of organisms. And I've been able to, and people in my lab have been able to use those data quite effectively. And so I have to thank at least some of the pioneers that got this thing rolling. And nowadays, genomes are a dime a dozen, but 10 years ago, they were actually quite important, and people that pioneered those, including Mitch Sogan, et cetera. And then finally, I have to, and at the end of my talk, I'll talk about this group of organisms that we're working on called rotifers, and my main collaborator is this guy, David Mark Welch, at Woods Hole, that has been important in this work. So let's talk about sex. I didn't time the music very well, apparently. So what is sex? Why do it? For other than the obvious reasons. And in particular, when and how did sex evolve? It's not obvious that this ought to be the case in biology. So we want to understand the evolutionary history, the natural history, and of course the underpinnings, sort of the molecular underpinnings of sex. And if we were able to, we might be able to answer this question. All right. So sex really is really sort of two halves of an equation. I call it the yin and yang of sex. And what we usually talk about when we're talking about sex is the getting together of gametes. Okay, so gametes are haploid, meaning they have half of the genetic material of all of us sitting out in the audience. We all have diploid, 2 copies of every chromosome. Okay, but we can all make gametes that are haploid, and those haploid gametes get together by fusion. And that's what we usually think about as sex, or the process that leads to the bringing together of those gametes. But the other side of the equation is that process that generates those gametes. And it's a complex molecular process, genetic process called meiosis, that takes those diploid cells, which we all have, specialized in our germline, the part of our organism that creates those gametes that are diploid, and halves them via this process of meiosis. And what I'm going to tell you about, which you might actually doubt is very sexy, but okay, is this process of meiosis and how it evolves as a way of understanding the evolution of sex. Okay? So what about sex? So the natural history of sex is interesting because it's quite variable. We generally think about sexual life cycles. Since we're humans, we think about our own sexual life cycles. And so we have a sexual life cycle in which we spend our lives as individuals as diploids, and we generate through meiosis haploid gametes, which have a very sort of, at least for men, have a very short lifetime, and they get together, and they don't make organisms themselves, but they get together and they fuse and they make a zygote and then make a new organism. Okay. In females, those gametes are actually sitting in meiosis at birth for mammals and persist for a long time. But anyway, so the main individual is this diploid individual, and it makes gametes. So we're used to thinking about this kind of sexual life cycle. But lots of organisms don't do it the way we do it. They do the opposite, which is to say that their body, if you will, are haploid. Right? They spend most of their time as haploid individuals, and then fuse. Those haploid individuals find each other and fuse and make a zygote, make a diploid individual, and then it immediately goes through meiosis and make another haploid individual, and that haploid individual persists as the sort of vegetative state. Okay. Some organisms actually can't decide, and they do a little bit of both. So like hornworts, shown here. So this was a fungus back here, but lots of different organisms do. So some organisms actually spend part of their bodies doing both haploid and diploid. So there's a lot of variation in terms of what parts of the body are diploid and haploid. Okay. So the asexual or growth phase, we think about asexuality as being the growth phase. Like in a real sense, our skin cells are asexual. They're just growing mitotically. Okay. They can be haploid or diploid or both. Okay. The other thing about sex is that we tend to think about gametes as being this sort of distinct thing, but in many organisms they're actually pretty much the same thing. They can be similar. They might have mating types such that there might be plus and minus, or there might be more complexity than this, but we call this isogamy, where you can't really tell the boys from the girls, as it were. Okay. We tend to think about this, which we call anisogamy, where gametes are different in some way or another. In particular, we tend to think of the big gametes as being eggs and the small gametes as being sperm, and that leads to the kind of thinking about the evolution of the sexes, which of course has happened actually many times in evolution, at least a few times, that we've had generation of multicellular organisms that make big gametes and small gametes, which we call egg and sperm, which have different roles in evolution. And so generally we think about males as having small, mini gametes, and their only function, really, sorry guys, our main function is fertilization. And females have big gametes, make relatively few of them, and their function is both fertilization and provision of nutrition for that early zygote. And it leads to this sort of idea, which is based in a lot of data, which is that in general, sperm is cheap and eggs are expensive, and that sets up issues in evolution about male and female, sort of a different kind of investments, okay, that sometimes happen in different kinds of organisms where this part of sex actually drives differences between the sexes in many ways. I'm not going to talk much about that. We can talk about it at the end if you want. What's next? Oh, yeah. So sex is really the rule. Even though there's all these differences amongst organisms, really sexual reproduction among organisms that we're going to talk about from here on, which are eukaryotic organisms that have nuclear, they have their genomes separated in a separate compartment called nucleus. It's the rule. Everybody's doing it for the most part. Some organisms need some help. And anyway, but it's widespread. Animals, plants, fungi, single-celled organisms like protists. There's some strange things like organisms that are hermaphrodites. It's actually not that strange in biology. We just think it might be strange, and et cetera. So there's lots of variation, but really sex is the rule. But sex being the rule, however, asexuality, the ability to make a clone of oneself among organisms, is actually quite a common feature amongst many organisms. Animals do it, lots of different animals, including even vertebrate animals, plants, fungi, protozoa, single-celled organisms, and this set of organisms, which I'll talk about at the end, which are rotifers, which are small microscopic animals, but they're actually thought to be the exemplar group in which sex is actually not known. So these organisms may have actually figured out how to live without sex. Okay, so they're all females. They only make babies by cloning themselves. They make daughters that are identical. I thought I'd mention just briefly here, you might be saying, okay, well, what about prokaryotes? So I was saying that really this talk is about eukaryotes, those cells with nuclei in them. Prokaryotes, bacteria, in fact, are asexual. Okay, but their genomes are set up in a very different way. They actually carry out a sort of sex in a different way. They actually exchange DNA sort of one gene at a time. When we're talking about sex, and we'll talk about it a little bit more in a minute, eukaryotic sex is about exchanging whole genomes worth of DNA. Okay, so meiosis, the process that I'm going to tell you about, is actually exchanging whole genomes worth of DNA. And so it's that innovation, that sort of complicated and a little bit confusing innovation that we're interested in its evolutionary history. So why sex? What's it good for? Okay. Another way of saying that in a scientific way, perhaps, is what is the adaptive significance, okay, of sex? There are probably many costs and risks that you can think of that are associated with sexual reproduction. So does anybody have any, this is where my teaching moment comes in, does anybody have any ideas about what might be a cost? Got some smart undergraduates in here, I think. What might be a cost of sex? Don't think about the good parts, just think about what might be the problems associated with it. Anybody? Yeah. Diseases, that's a good one. It's on my list, it's going to show up. Any other good ideas? Is it easy? Oh, Cindy's got an idea.
You have to find a partner.
You got to find a mate, right? A partner. It's not always easy, right? Match.com is out there for a reason. All right. Any others? All right. Searching for a mate, time and energy, and a good computer sometimes. That mate actually might require some investment, right? Sure, you love each other, but is your credit compatible? Risk of sexually transmitted diseases? Not without risk. And a fun one, which is that, you know, sex in and of itself is not always safe, depending on what kind of organism you are. You might be familiar with the case of the praying mantis, and so this is a female praying mantis, and this male praying mantis is sneaking up on her and attempting to mate with her, because if he doesn't sneak up just right, she's going to bite his head off, literally, and eat it. And, you know, he'll continue actually mating with her even with his head gone, but that will assure his paternity in the future. So sex isn't entirely without cost. So why do it, right? Like I said, asexual reproduction is actually pretty widespread across eukaryotes. So why have an organism just given this up altogether? Well, a very famous evolutionary biologist, probably the most important evolutionary biologist of our generation, of the last century, John Maynard Smith, spent a lot of time talking about this and thinking about it and writing about it. And he asked, you know, in populations that can do both, why in the heck are they doing sex, right? Because asexuality is much easier. And in fact, it's not only easier, it's obviously easier, because in a sexual population, at every generation, the mother actually, in producing male offspring, is reducing her output, right? So by combining her genes with another organism, she's having her genetic contribution and reducing her own contribution to the next generation. In an asexual population, the mom can make, this mom's making 2 organisms, one of which is a male. He's not good for much of anything. But here she can make 2 daughters that are identical copies of herself. So in terms of evolution, in terms of bringing your own genes to fruition in the future, this is pretty costly, and you can see that sexual reproduction loses in this game head-to-head. So Maynard Smith didn't, and this comes from Wikipedia. I don't always point to Wikipedia, but this is a Wikipedia page that's pretty good. Okay, so given all these things, you know, all those bad things about sex, then why aren't most organisms asexual? Because under those just sort of simple ideas, and they've been fleshed out in some detail by lots of evolutionary biologists, asexual reproduction should win, right? Once it evolves, if an organism can do both, they ought to just go completely asexual. Okay? But it's not the case. As I showed you, sexuality is widespread, what I didn't tell you is that, in fact, most organisms reproduce sexually. And in fact, many organisms can do both, right? So many organisms can switch between sexual and asexual reproduction. So given that they can switch, why don't they just always go asexual? So that must mean that sex in and of itself probably provides some benefits that are going to overcome whatever those disadvantages, including some others that we didn't mention of asexual reproduction. Okay? Questions? So evolutionary biologists have been thinking about this for a long time, actually for more than a century. And so there are a number of theories for why sex, what we call maintaining, why sex is being maintained, persisting. There must be some reason. There must be some good reasons for sex to be maintained because it's obviously suboptimal in many ways. And I call them the good, the bad, and the ugly. Okay, so the good, it might make some sense to you, is that one idea for the good is that sex creates variation, right? Sex that creates variation and it's beneficial for adaptation, and Weismann at the turn of the last century put forth this idea, and then later on Ronald Fisher put forward the more explicit idea that sex actually allows the bringing together of good mutations in two individuals that otherwise might not see each other if it's asexual. Okay, so in an asexual population, they're never going to see each other if good mutations come about, but if you can bring them together every once in a while, that would work. So there's Ronald Fisher. The bad, put forth by Muller, is that sort of the flip side of the good is that deleterious mutations will actually accumulate in an asexual. So think about a Xerox copy, right? You make a Xerox copy. It looks pretty good the first time. You make a copy of the copy. It looks pretty good. You make a copy of a copy of a copy. It might start looking less good. And so that's the idea of the bad mutations. They sort of build up. And we call it, and in fact, this is such a clear idea that it's actually been called Muller's ratchet. So once you're down to the 100th Xerox copy of the Xerox copy, you can't go back to the original. You've all seen these when in lecturers that like have copied their notes for 20 years, right? I'd never do that. All right. So that's Hermann Muller, who won the Nobel Prize actually for mutations. Actually, so he, bad stuff was sort of part of his gig. And then the ugly. So I call this the ugly. And one specific idea with the ugly is that sex is good to sort of adapt to changing environments. And the ugly part that specifically Bill Hamilton, I think, spent a lot of time talking about was parasites. So your parasites are evolving with you, and you are evolving to beat your parasites, and then your parasites keep up with you, and so you're constantly evolving. It's called the Red Queen hypothesis, and Bill Hamilton was a major part of this idea. So basically, there's all these theories, none of which have sort of taken complete hold of evolutionary biology. It might be some combination of them, and we don't. This is sort of an open question in evolutionary biology. Which one of these things, if any of them, are actually the explanation for sex? And I would suspect that probably none of them are the explanation, but some of them in combination might be provide explanations depending on what organisms we're talking about. Okay, so that's all the context. So now I'm going to tell you a little bit about meiosis. So that was the sex part. So now I'm going to give you a little bit of an overview of what I'm going to talk about that's research in my lab. So asking questions about when meiosis, and thus sex, arose. How did meiosis arise? And I'm going to try to answer these questions to some extent. So meiosis is a process that's a new process that arose sometime during evolution. We know that evolution works by taking advantage of previous existing functions and putting them into new jobs. So we're asking, what prior functions? What are those functions that pre-existed that got usurped, that got called into play for meiosis? Given that meiosis is there, does it stay the same? Has it always stayed the same? Or how has it evolved? A question that we're really, really interested in is, once it's evolved, once it's there, is it possible to lose it? Are we stuck? Okay? And then finally, a question which I'm going to tell you right now I'm not going to be able to answer, which is, what are the consequences of understanding all this stuff in this persistence, this maintenance of sex? I think there's something to be said in the future about this, but I and it motivates the work, but this is a very deep question in evolutionary biology because it remains an open question. All right. So I'm going to give you a little bit of a framework for the kind of work that we do, and this is a phylogenetic tree. Okay. So this is a tree of all life. And life on this planet basically can be broken up into 3 clumps. There are 2 kinds of bacteria, what we call eubacteria. These include things like E. coli and Salmonella. If you've gotten sick from food poisoning, you might have gotten Salmonella. And Archaebacteria, which probably you've never encountered because none of them are pathogens, but they're prokaryotes. They're single-celled organisms. They're in many ways similar to eubacteria, but they're quite distantly evolved. And then this big clump of organisms we call eukaryotes. These all have nuclear genomes. They all have linear chromosomes. They all have cells like we have. A vast majority of these things are single cells. Okay? They're single cells. They're protists, protozoa, amoebae, things like that. So you have to, they're very tiny, but they're actually much bigger than prokaryotes because they actually have that complex machinery that defines a eukaryotic cell. Things like organelles, mitochondria, stuff like that. Okay? So a vast majority, all those white lines, are all single-celled, what we call protists. The organisms that we're most familiar with, animals, plants, and fungi, the big ones, are in some ways sort of latecomers on the scene of eukaryotes. So eukaryotes have been around for a really long time, and we think there's pretty darn good evidence that eukaryotes, complex single cells, evolved from less complex, smaller prokaryotic cells. So that there's a little bit of controversy, but for the most part, the data are quite consistent with eukaryotes being sort of an advanced version of a single cell. And so when I started thinking about this problem, I wanted to ask, well, among the eukaryotes, who's having sex? Okay, who's doing meiosis? And it turns out that these organisms, diplomonads, trichomonads, and microsporidia, diplomonads, trichomonads, and microsporidia. Has anybody met Giardia? Raise your... Oh, somebody's met Giardia, sorry. If you're laughing, you know. So, Giardia is that thing that you are afraid of when you go backpacking. You have to filter your water, or else you're going to get sick. These are organisms that looks like, based on this phylogenetic tree that's based on a gene that everybody has, might have diverged early in eukaryotic history. Okay? The cool thing about these organisms, at least 10 years ago, was that they weren't known to have meiosis. People have been looking at them. These aren't common words amongst our parlance, amongst our breakfast conversations. So people haven't really studied these very much, but people have studied them enough to have looked for things like meiosis among these organisms, and they didn't have it. So it suggested early on that maybe these organisms never had it. They were eukaryotes. They had nuclear genomes. In fact, these little things that look like eyes are actually nuclei. They have 2 nuclei instead of 1. There's a nucleus. Okay? They're clearly eukaryotes, but they seem to not have sex. So it suggested that maybe sex is something that came along later during eukaryotic evolution, right? It got invented late. That would have been great, except for when there are 2 things that happen. First of all, we begin looking at constructing these trees of life from other kinds of molecular markers, including proteins, and all of a sudden those organisms that seemed to branch early in eukaryotic evolution didn't. Okay, science moves on. The other thing is that we started learning more and more about the presence of meiosis amongst a wider variety of taxa. So opisthocons are animals and fungi and some protists, some single cells. Amoebozoa, these are all amoebae. Plants are plants. And some green algae, some other protists, some protists, and some protists, and some protists. Okay? So most of eukaryotic diversity are single-celled protists. Okay? But it turns out when you sort of think about the fact that it didn't look like things like Giardia branched early, plus the distribution of sex along this tree was a little bit more complex, it led people to start talking in about 2004 that this last common ancestor, so these are all prokaryotes, eubacteria and archaebacteria, that this common ancestor of all eukaryotes was pretty much a eukaryote. It had a nucleus. It had organelles called the endoplasmic reticulum and the Golgi. So these are organelles that move things around within cells. And it underwent 2 important processes, mitosis and meiosis. Okay. We went on a few years later to sort of show this by looking at lots of genes, and I'll show you some of those data. But that's actually the answer. Is that this initial answer turned out to be wrong? That meiosis, in fact, was a process that defined early eukaryotic evolution. All right. So let's go back just for a minute and talk about what we know about meiosis. So there's that blue slide that I showed you before. There's all those eukaryotes up here. And all I've done is taken this portion and splay it out on the side, okay? Just to show you, most of these things on here are protists. And there you are with your fungal friends, okay? So animals and fungi are just this sort of twig on the tree of eukaryotic life, okay? And the reason why I show you that is that what we know about how meiosis works comes almost entirely from that part of the tree, okay? So what we want to understand about how meiosis evolves is by taking what we know here and then spreading it across the rest of the tree by looking for genes that are shared that carry out that process. So this is now blowing up that little purple part. So there's some fungi, things like Saccharomyces cerevisiae. It's called baker's yeast, but we all know what it really is used for, brewing beer. Anyway, various fungi, including mushrooms. There we are, humans are pretty close relatives of the mouse, and Drosophila, fruit flies, and worms. We have a worm researcher in the audience. And so what we know about how meiosis works comes from a lot of different animals, plants, and animals and fungi, and a little bit from plants. But still, that only represents a little bit of the tree of life. Okay? Is everybody following? These trees are a little bit complex, but I'm trying to don't think about the details. Just sort of try to follow the generalities. So now I'm going to show you a little bit that splayed out version of that tree that I showed you before, where there's animals and fungi and some protists, and there's plants and some protists, and then these are the rest of those protists that I've got in here. And what I'm showing you are boxes around animals and fungi and plants to demonstrate that those are the groups of organisms that we know something about how meiosis works. What are the yellow boxes? The yellow boxes represent organisms where we have genome sequences. And so what we want to do is we want to take what we know about genes that are shared, that we know work in meiosis in animals, plants, and fungi, and we ask, are those genes also present in those other organisms? And that will tell us something about whether meiosis is present or how meiosis might work in those organisms. Okay? So that's what we've been doing in my lab for a long time. This is a little bit out of date. We actually have some genomes in this group, actually quite recently. We've only been mining them in the last month or two. But most of the tree of life has at least one or a few genomes. Okay? So we can actually begin to ask across the whole tree of eukaryotic life, who has what genes? Who has what parts? Okay, that's natural history, right? Going out and collecting and putting them in different categories and comparing them and learning something. Okay? That's what we do. But we do it for molecules. So a little backtracking. Let's think about what we knew going in. I sort of told you that we didn't really know that much about who's having sex across the eukaryotic tree. Most animals have sex. There's a lot of asexuals, but for the most part they're having sex. Fungi, this group of organisms I mentioned early on, those early diverging things, turned out they're actually relatives of fungi. So that's one interesting thing. But most protists, these blue things, these things labeled in blue, we either don't know anything, or in some cases people have looked and there's nothing there. Right? They just haven't found any evidence. So what would the evidence be? Well, obviously catching them in the act, right? The other way that we do it actually is by doing population genetics. We can go out and look among individuals in the population, and if they're having sexual reproduction, they sort their genes in a particular way that will tell us something about whether they're having sex or not. But just because we can't catch them in the act, or we can't find evidence for this, does that mean that they aren't having sex? It doesn't actually. It just means that we might not have found the right conditions. I always like to say, maybe we haven't got the music right. We don't have the lights down low. We don't always know what the lab conditions are to get organisms to have sex. So this is what we call absence of evidence. Okay? And so when we have absence of evidence, things like minuses here, right? Is that indicative of asexuality, or are they just doing it on the side in ways and in times that we aren't paying attention? Okay? Or very, very infrequently, which actually does happen in many organisms. So we have to be really worried about just going and observing sex and saying, well, we didn't observe it, so therefore they must be asexual. And that's a big part of what we do in my lab, is sort of to get over that problem. So we look at meiosis. And so now this is the beginning of a kind of, we're all going to go, okay, meiosis, I might remember that at one point. It really confused me. So meiosis is actually a very cool, and this is my University of Iowa meiosis slide, obviously. Meiosis is very simple and straightforward. I hope I can demonstrate that. So it comes in two different divisions. It starts pretty much the same way as mitosis. Chromosomes replicate. But what happens is that instead of going on and just splitting and dividing, they go through a special phase here, which involved recombination. Okay? And then they go through another division, but they actually don't replicate. And so what happens in the end is that they end up halving. Okay? They go through a reductive division. Okay? Creating those, you start from 2N and you go to 1N. Okay? So that's what meiosis does. Meiosis 101. So compared to mitosis, it involves that same replication here, but instead of the chromosomes lining up and then separating, they pair up with one another. And dad and mom chromosomes, so my daughter here has chromosomes from both my wife and I. Sorry, I'm going to embarrass her just for a minute. So it's not quite as fun as that. There's actually all kinds of complexities in meiosis. There's a bunch of proteins that do jobs. So there's those newly replicated chromosomes, and these different colors of blobs are different proteins that carry out different functions. And I have the sort of functions listed over here, including proteins that bind chromosomes together, proteins that cut DNA. These are involved in that recombination, so chromosomes swapping DNA with one another. It's actually started by a cut. And then there's a bunch of proteins that carry out various aspects of recombination. The neat thing is that meiosis, so you're like, okay, well, DNA is important. Why are we cutting it up and pasting it back together? It turns out that many of the proteins involved in meiosis are actually also used in other aspects of what we call DNA repair. These are proteins that fix DNA when it's broken. But meiosis works by also including some proteins that are very specific to meiosis, and these are the green ones shown here. And these are proteins that are carrying out those very specific jobs of meiosis and meiosis 1. And we're interested particularly in studying those. So in order to study the evolution of meiosis, we wanted to actually look at genes that are meiosis markers. We want them to be specific to meiosis. The reason why we want them to be specific to meiosis is because those genes tell us the most about how something evolves, because if meiosis isn't there, there's no reason for those genes to be there, because they only do that function. One thing I forgot to tell you is those trees of life, we're talking about 3 billion years of evolution. And eukaryotes are about 2 billion or so of that evolution. So the genes that we're interested in have to be slow enough evolving, conserved, to study over that long period of time. So these are proteins and genes that are actually, whose sequences are actually really, really important to what they do, to what their jobs are. And I won't be able to tell you much about gene duplications. So as a molecular biologist, when we want to study this process, we want to have some genes that everybody who's doing sex has those genes, having meiosis. And if there are any organisms that are not having sex, those genes ought to be gone. Use it or lose it. That's a rule in evolution, right? If there's no need of the function, those whatever processes are carrying out the function disappear because there's no need to carry them around. And so those genes ought to be absent. They ought to be gone, or at least in the process of being gone. Does everybody get that? Cool. All right, so we look for presence and absence of genes to tell us something about whether organisms are carrying out meiosis. Okay. So meiosis, as I said, is this process that's carried out at the DNA level and is carried out using a series of proteins. And it turns out that many of these proteins are also involved in fixing DNA, proteins that fix DNA. And I put boxes around some of these genes because they are close evolutionary relatives to genes that are actually specific to meiosis. They've evolved by what we call gene duplication, that one gene made another copy of itself, and now it does a specific function in meiosis. And it's those genes that we're particularly interested in. So many of these genes have evolved by what we call these gene duplication. I'm going to show you one example of this that we've been studying for a number of years. And that's called RAD51 and DMC1. And these are proteins that are involved in that exchange of DNA, okay, among chromosomes. And bacteria have genes that exchange the DNA, and they use it to fix DNA. But eukaryotes have one that's used for mostly fixing DNA, but they also have this thing called DMC1, which is specifically involved in that recombination aspect of meiosis. In fact, it's one of the proteins that directs the recombination to go from the chromosome of dad to mom or mom to dad, so that you actually get those recombinant, you get those crossing over events. It turns out that the phylogeny of these things is such that there was a duplication, and the way that you look at these trees is that these are prokaryotes, and that common ancestor had a REC-A gene, had a single gene, and that at some time during eukaryotic history it split. So now there's two genes. There's this meiosis one, and there's this general one. And animals and plants and fungi have both of them. And what we really wanted to know is, what about everybody else? The rest of that broad set of eukaryotic diversity, we wanted to know, did they all have this and this, or were there organisms that branched before those things got invented? Because this is specific to meiosis here. And if this organism branched off the tree of life before this gene got invented, that might tell us something about that organism never learning how to have sex, right? So that's sort of how we think about this thing. It's a sort of simplistic way of thinking about it, and so this is a basis for how we do the work. We are molecular biologists, and we go into genomes and we pull out genes using this technique called Degenerate PCR. We've had some Degenerate PCR mavens here in the audience, people in my lab that do this. I won't explain it except for to say that we use conserved sequences to design DNA probes to go into organisms and clone their genes and sequence them. By doing that kind of thing, we can actually clone and sequence genes from organisms, and what we found was that DMC1, this meiosis-specific thing, everybody has it. Everybody also has this, and we expected that. This is our control, which led us to sort of fill out this hypothesis in a way that says everybody's got DMC1. We never were able to find this thing. That's consistent with there never being an organism that diverged prior to the origin of meiosis, at least one that has persisted to our time. There's an interesting case in this Giardia. They actually have lost this general one, but I won't have time to talk about that. Okay. We keep doing this. So this is not even that recent of a tree from my student Art Peitling, where we've filled in lots and lots of protists. So the protists are in blue here, animals, plants, and fungi, and everybody has DMC1 and everybody has RAD51. Not everybody has DMC1 because in some cases, as I'll show you, they've been lost. But their origin predates the evolution of meiosis. Okay? Just one gene. I could tell you all kinds of stories about all kinds of different genes, but I'm not going to do that today. I'm just going to tell you a little story about one of these genes that gets back to that video. So we have stories about genes involved in DNA breaks, genes involved in crossovers, genes involved in various aspects of chromosome mechanics. Increasingly, we don't do that degenerate PCR stuff all the time, although we do it a lot in my lab. We actually go out to databases, and people are sequencing genomes, and we run through this sort of complex series of computer manipulations to go and find genes, find out if they are what we think they are, look at them carefully, annotate them, which means sort of figuring out whether they really are what we think they are, putting them in those phylogenetic trees to figure out what they are, and then assessing them as to whether those genes, in fact, are these meiosis-specific genes that we think they are. I'm going to tell you a little story about this particular set of molecules. So these are those, remember when that, at the beginning when that chromosome replicated and there were two, okay? These are proteins that, a protein complex that sort of hold those things together are called cohesions. You see they're kind of ring-shaped, they hold them together. These work both in mitosis and meiosis, but in meiosis there are specific components that are close relatives of the proteins that do this. And I'm going to tell you a little bit of story about this one called STAG3. It turns out that, and so this is a phylogenetic tree of those proteins, and it's a little complicated except for to say that there are 3 versions of this gene in vertebrate animals. So this tetrodon is a fish, and human, rat, and mouse. And you can see that there are 3 different versions. And there's a one that only works in meiosis called STAG3. There's another one in fungi that also is specific to meiosis called REC11, but not all fungi have it, just this one. And there's not a meiosis-specific version here. So this is a case where a meiosis-specific version of a protein evolved independently multiple times. I'm waiting to tell you about this one. So Drosophila, fruit fly, also has a meiosis-specific version, but what's really interesting about it, it actually got named S&M. Okay? And the reason why this protein got found and this gene got found was because Drosophila does something really funny with its meiosis. Does anybody know? Biology professors can't answer. And TAs. Any undergraduates know what's funny about meiosis and Drosophila? Cindy? Anybody? It gets to why that film is wrong. In Drosophila, males don't recombine. Okay? They've figured out how to actually segregate their chromosomes without actually going through recombination. Okay? And so it turns out that this is a protein that is specific to how meiosis works, but it only works in males. Okay? And so, so we began studying this, and so this meiosis-specific thing evolved. And this is quite recent because this is mosquito, and they're actually quite close relatives of fruit flies. And so what we wanted to do is ask, well, if that's the case, then when did this special kind of meiosis, this weird meiosis called achiasmatic meiosis, or chiasmata being crossovers, so there's a meiosis that doesn't involve recombination. And so there's Drosophila. And up here are mosquitoes. They actually aren't on here. But there's a wide diversity of flies. And what we wanted to ask was, and so we actually are working on a bunch of these different flies, and this is just some nice pictures of the organisms we work on. We wanted to ask, when did this new gene that is specific to meiosis, when did that evolve? Did it evolve somewhere in here? Okay? We didn't really know about meiosis and its distribution. So we do this degenerate PCR business, and we pulled out lots and lots of sequences. And what we found, to our surprise, is, so here are all those insects, all those dipteran flies, flies with two wings. And the specific version to meiosis is only present in Drosophila. Okay? So it's a really recent invention. So my student Danielle has been looking even more carefully, and still only found in Drosophila, among the genus Drosophila, lots of species of Drosophila. But you can see there's a whole bunch of different dipteran flies in here that don't have that. So it's a relatively recent invention in fly evolution, including Musca housefly doesn't have it. So so meiosis isn't set in stone, right? Meiosis has changed. It's actually changed quite recently. Okay, and so we're trying to figure out even more narrowly sort of when that happened. So that's just one little story. So in general, what we're doing is we're taking this natural history, this inventory, as it were, of all these different genes. Now this is getting really technical, but I'm going to try to generalize it. So each column here in yellow are genes that are specific to meiosis. The white columns are genes that we expect to be there anyway. So organisms like Giardia and Trichomonas, these are those organisms I talked about before. These are all organisms in boxes that weren't known to have a sex life. Right? I told you one of the reasons why I was interested in them because they weren't known to have sex. Maybe they never learned how to do it. But look at this. They have this meiosis-specific gene, all these, etc. So these are organisms that weren't known to have sex, but yet they've got the parts, as it were. Okay? And we found over and over organisms that weren't known to have sexual reproduction, this absence of evidence that we could find that they had a lot of these genes that we expected to only function in meiosis. And we looked at this even more. So now I'm going to show you sort of an increasingly complex series of slides just to show you that some of these organisms, like us and yeast and other fungi, it turns out there's a lot of these genes. This is now a survey of about 29 genes. It's not that single cells have on a lot fewer genes than us. In fact, Trichomonas has 27 of the 29 genes in this survey that are involved in meiosis. So there's a series of genes that predates all eukaryotes. They're present among all eukaryotes. But these empty boxes here probably represent cases of genes that have been lost. So meiosis hasn't stayed exactly the same. It's been evolving by new genes, like in Drosophila, gene loss in some of these cases here. Okay. So this is sort of coming to a point, two things I want to tell you. Number one is what I just said, and a lot of stuff that I've talked about so far suggests that meiosis evolved very early on at the base of eukaryotic evolution, once. a wide diversity of eukaryotes has that set of machinery that defines meiosis, and that asexual organisms that we thought were asexual are actually sprinkled all along the tree, these number 2s. They're all over the tree, which suggests that if so, it wasn't that they never learned to have sex, it's just that they probably are capable of sex. Okay? They have the genes, or else they lost the ability to have sex very, very recently. Okay? Can we use this natural history? We know, for the most part, who has what genes. Can we use it as a way of going into an organism that we don't know is capable of sex to ask whether they're capable of sex? And what we really want to know is everybody doing it. This question, can meiosis be lost? And there's lots of asexuals that we can look at. And this gets to that deep question about the presence of sex. Is it actually a necessary feature of being a eukaryote? All right, so this is a fun part. We have a putative asexual. When I went looking for putative asexual, I'm a biologist. I know about lots of different asexuals, but of course I went to Google, and I said, put in asexual organism picture. You don't want to know all the things that showed up, but one of the things that showed up was kind of surprising, and it stuck. I mean, is that according to his creator, SpongeBob is asexual. I mean, it doesn't actually make sense that SpongeBob is asexual. Maybe SpongeBarb. Okay, because, boys can't really be asexual, okay? But anyway, okay, so SpongeBob is asexual, says his creator. But we don't really believe that SpongeBob, we want to test, we're scientists, we want to test whether SpongeBob or any other asexual organism that we're interested in. The traditional way would be we'd go out and look for SpongeBob populations. We'd go out and do population genetics. We'd go out and gather different individuals, and we'd grind them up their DNA, and we'd ask whether they're recombining. Okay, but you know that that's not what my lab's going to do. We're going to ask this question. We're going to say, do they have the goods? Right? If they've got the goods, they're doing the deed, right? If the meiosis genes are there, we might go back and do population genetics. That's hard work. We have to go out and do collecting out in the field and stuff like that, and we're lab rats in my lab. Okay. Anyway, we're going to have these let's say SpongeBob has a bunch of these meiosis genes. We want to ask which ones, how many? Okay? Because if so, then we could start asking questions about their function. Are they expressed? Are they turned on? Are they starting to make functional products, right? RNA. If they're expressed, we want to ask when and where. We know, as on that slide that I showed you, that there are certain steps in meiosis. The DNA breaks ought to precede those proteins that are involved in the crossovers, for example. We know that the order of events ought to be more or less constrained. They ought to all be in the same place in SpongeBob. If one gene is expressed here, and one gene is over here, and one gene is over here, then that's maybe not so good. But if they're all expressed, say, here, or maybe in his ear, because different organisms express their germlines in different parts of their bodies, in the same place, let's say, that might be good. A little bit more complicated because SpongeBob is not a genetics test system. It's not a Drosophila. We can't grow it in the lab very well. So we have to have a way of assessing them, and we can drop them into things like yeast cells as test tubes. If all these things look good, we might then be able to get a granting agency to say, we really want to get SpongeBob to do sex. We really want to understand it, right? So can we induce it? I mean, if SpongeBob starts out as being an asexual, the granting agency is going to say, why would we give you money to induce sex? We know that SpongeBob doesn't have sex. But now we've got all this evidence. Can we induce meiosis? If so, he's not an asexual, and we, So, all right. So that's essentially the global view of the kind of thing that we're interested in doing in my lab, is sort of looking for organisms that are asexual and then asking these kinds of questions. And these are our test subjects, at least initially. We've got some other ones. So these are the girls. These are deltoid rotifers. These are female lineage that have persisted for tens of millions of years. Some people say up to 100 million years, which is a really long time. Maybe 40 million years. So there she is with her daughter, who is genetically identical to her. Okay? And it's a group of organisms. They're microscopic. You can come up to the lab and look at them sometime. And their closest relatives, which are called monogonate rotifers. And these rotifers are also capable of making clones of themselves, but they're also sexual. So they can actually switch depending on conditions. And so we're going to compare what's known about these with these and ask, what do we know about the meiosis genes here, and can we find the meiosis genes in these girls? Okay? Or are they all girls? Turns out that these have been pretty well studied. They're quite famous. There's no evidence for males. People, Leeuwenhoek, like one of the famous people that developed microscopes, talked about deltoid rotifers and found no males. They're old. They don't have these homologous chromosome pairs that you would expect for sexual organisms, et cetera. There's a whole bunch of things that are consistent with them being asexual, but maybe not sufficient. So the genetic evidence is consistent with, but they're all based on these consequences. But what we really wanted to know, and now I'm sounding like a broken record, do they have the capacity for sex? Do they have the goods? Okay? Do they have the meiosis-specific genes? If so, our easy hypothesis is they're probably doing the deed. If not, it's a win-win situation. Then that's a really interesting case, and that's why Maynard Smith promoted these as organisms to study. If we can study the exception, then that might tell us something about the rule. We have genes that I've told you a little bit about. They're meiosis-specific, they're conserved, et cetera. Okay? We go look for the genes. Degenerate PCR, I didn't get to really tell you about it. This is Andrew Shurko, my longtime postdoc. Easier said than done. True? Cindy's nodding her head. Yeah, it looks easy, but it's actually not that easy. But luckily we've had data from the monogonons. So these are the sexual organisms. The meiosis-specific genes are in pink, and then the ones that everybody's expected to have are in black. The monogonons, the sexuals, we have a genome for these two things. They've got all the goods. All right. We've been doing degenerate PCR on some of these, and you see that it's spotty. It's hard to do, actually. Just because we didn't find it doesn't mean it's not there. But what's really interesting is that we found meiosis-specific genes among these red organisms. These are the deltoids. These are not known to have sex. What the heck are they doing with those meiosis genes? Right? A little bit different way of looking at it. There are the genes. The meiosis genes are in yellow. The deltoids are in blue. Meiosis-specific genes, et cetera. Okay? So we have evidence for these genes that we think are only involved in meiosis in lots of other organisms. We don't know whether they're only involved in meiosis in these organisms, in these deltoid rotifers. So what does it mean? Simplest explanation is they're doing it on the side somehow. They're clandestine. All right? Maybe they're there, but they're not turned on. One interesting thing that happens in evolution is that sometimes genes, and I talked about the origin of meiosis itself, genes gain new functions. All right? It's harder to explain for many, many genes. For one gene, it might be okay. Look back here. We've got a number of these genes that are doing different things. So that's a little bit harder explanation, but possible. An interesting thing for the aficionados, meiosis is present, but it's used for automixis, which automixis is basically sex with oneself, right? So you make gametes and your own gametes fuse. You get some benefits from meiosis that way, but eventually those benefits go away. One possibility is that they might be not rotifer genes. They're actually coming from some other organism that's either contaminating or that they've eaten or something like that. We can pretty much rule that out with the kind of methods that we use. So anyway, we prefer this hypothesis, but we're not quite there to actually nail it down, so stay tuned. All right, so I'm going to answer the questions. When did meiosis arise? Very early. How did it arise? By lots and lots of gene duplication. I didn't actually show you those data, but it turns out that new genes don't come out of nowhere. Most of these meiosis genes evolved from other genes, from functions that have a role in fixing DNA. How has it evolved? There's lots of gene loss, those holes in those slides. Some new genes and a little bit of more gene duplication. Okay? Don't know the answer to this question. If meiosis can indeed be lost, they would be really interesting organisms to look at. Okay? And this bigger question, that's a career question, so ask me when I'm an emeritus professor. Okay, thanks.